Alright, this is our second video for section 6 of chapter 8. In that first video we talked about operons of prokaryotes and how they can turn their genes on and off by controlling that transcription itself. In this video we're going to talk about how gene expression is regulated in eukaryotic cells. Okay, so as you can imagine, expression in eukaryotic cells is much more complicated because you're a lot more complicated than a bacteria is. Most regulation does, again, have to do with transcription. So just like before, we're going to have some of those operon systems, just like the prokaryotes did, where you can turn it on when you need it, turn it off when you don't. But other things for eukaryotes, right, and to be honest, with eukaryotes, again, we're really complicated. We don't really know that much about how we express our genes altogether, right? We're still learning that because we have so much DNA and we don't even know what all that DNA. We've just, in the last 10 years, discovered the code for all of that DNA. We haven't even begun to sort out what all of it does and all the different proteins that you make. So we don't know a whole lot, but we do know a couple extra things. And again, it's just a little bit more complicated. One thing that's not very complicated that I would like you guys to understand is that we have promoters in our DNA that helps RNA polymerase recognize the start of a gene. And more specifically, I want you guys to understand how that promoter works. Right? It's called a TATA box because it would have this sequence of letters. And as you can see, it's all T's and A's. And if you guys remember from before, A goes with T, G goes with C, but between A and T, there's only 2 hydrogen bonds. So if you have a bunch of T's and A's in a row, and their complement is also a bunch of T's and A's, then there's not a whole lot of hydrogen bonds holding together that little part of the strand of DNA. And that actually helps the RNA polymerase pull those 2 strands apart in that specific spot. So we're looking for promoters because that would be the start of a gene, right? And it's really convenient because there's only 2 hydrogen bonds. So it's easy to separate the DNA right there. That last note's on transcription factors binding to specific parts of the DNA, not only the promoter, but also enhancers that could be really far away from the gene being regulated. Basically, that's talking about how there is actually some folding and bending of the DNA, and it can influence itself. And it's really that stuff is really kind of complicated. And so we're not going to worry about it too much. So understand that, again, eukaryotes, we're a little bit more complicated than those prokaryotes. Mostly we're going to worry about trying to understand those operons because those are actually complicated enough as is. But also just add in that promoter for a eukaryote, that TATA box, and understand why that TATA box would be useful. It's easy to pull the DNA apart there because there's not as many bonds between those bases. But another thing, as far as regulation is concerned for eukaryotes that I would like you to understand is that not all of that mRNA that's made is actually used. That RNA is processed before it's turned into a protein, generally speaking. So we're going to have these 2 different things. We're going to have exons and introns. Exons are the part of the RNA that code for a protein. Introns are just the parts that are cut out that are non-coding that don't actually do anything. So before you can actually read a piece of mRNA, you have to get those introns out of there. Otherwise, it all comes out as gibberish. So mRNA has to be processed first before we can make our proteins. So I got a picture to go with that here. This is just a piece of mRNA. And we're assuming here that A, B, and C are all exons. So those are the parts that can be read. Those are the parts that can be translated to make a protein. Whereas those other parts in between are introns. They need to be removed in order to make this thing useful. So you just cut those out in that processing. And you end up with a smaller piece of RNA. Let me use red again. Change pen color. Use this. You end up with a smaller piece of RNA. And the other thing that you should understand is that A, B, and C don't necessarily have to go in that order. I could actually put B first, then A, then C, or lots of different combinations, and that can actually lead to lots of different proteins. But I'm just going to splice those pieces back together after I remove those introns. And now that piece of mRNA can be translated to make a protein. Right? At the end of the day, that's what we want. That's what we need, that protein. But before that mRNA is processed, we can't get the protein. So it's just another form of how we can regulate that expression of our DNA in eukaryotes. So we're going to process that mRNA, cut out the introns, put those exons back together, then you can make a protein. The other little note on this slide over here was that you're going to have a protective cap and tail. They're going to be added onto that mRNA. You just want to protect it at the end of the day. And what that looks like, right? The cap on this side, I'm trying to figure out what color I use. The cap on the 5' end, right? Don't really worry about what that looks like. It's just a protein complex that's attached there. But the cap on the 3' end, right? All that's actually done is instead of just ending that nucleotide sequence there, I'm going to add a bunch of new bases here, but they're all the same base. They're just a bunch of adenines add to the end. So it's called a poly-A tail because you have many A's added onto the end. And basically, again, the protective cap and that poly-A tail, the whole purpose is that now I have protection on either side. Remember, the ends of any piece of nucleic acid is where it's most likely to get damaged. So I want to protect that RNA so that it doesn't get damaged or destroyed before it can make a protein. So again, that's that last little bit there. You got to protect it, remove the introns. You can actually put B, A, and C in together in different orders to make different proteins. But all of this processing has to happen before you can make that protein. So it helps us control that expression.